All right, AMP1 students, we are going to talk about myelination, and specifically we'll talk about how it happens and what myelin is actually made up of, what's its components. So first, myelination, when we say that, we're just talking about that process by where part of the axon gets wrapped in myelin. And so myelin is that material that insulates the axon and allows for faster conduction of an action potential or nerve impulse down the axon. So the myelin is primarily lipids, made up of lipid molecules. There are some proteins too, but it's mainly lipids. So because it has that high lipid content, it appears white. So when we are looking in lab at the brain and the spinal cord, when we cut into the brain and the spinal cord, I think it's on here, no, wrong one. Oops, up here. When we cut through the brain and spinal cord, we will notice, this is of course the brain, we'll notice that there's some areas that's white and some areas that's a darker color. So they call this area white matter, and they call the more peripheral outside that's a darker color, they call that the gray matter. And so the reason that we have this area that looks so much more white is because it's full and it has a high concentration of myelinated axons, or axons with myelin sheaths. And so that white color comes from all the high lipid content of the myelin that's surrounding all the axons. And so in the gray matter area, we don't have a lot of myelin. We mainly have cell bodies and synapses in that area, and we don't have axons with a bunch of myelin. So it's kind of the reason why we have gray matter and white matter, that we can see in our brain and spinal cord. Okay, so back to our textbook on myelination. The other thing that our objective is to understand the process of myelination. And so if we're talking about a neurolimocyte, also called a Schwann cell, how it produces a myelin sheath is that we have this neurolimocyte. So here it is. It has a nucleus. What it does is here's our long axon, or at least a section of it. What it does is it'll fold around the axon, kind of like a hot dog bun around a hot dog. But it doesn't stop there. It'll continue to wrap itself around and around and around that axon until you get this thick layer here, and that's the myelin sheath. The whole thing is a neurolimocyte or a Schwann cell, but the layers that it produces around the axon is called the myelin sheath. So remember, our Schwann cells or neurolimocytes, they form myelin sheaths in the peripheral nervous system, and they can only form one myelin sheath. Whereas the oligodendrocytes are the cells that form myelin sheaths in the central nervous system, and they can actually form more than one myelin sheath. Okay, so now we're going to talk about, let's go to our objectives, make sure we don't forget anything. We're going to talk about multiple sclerosis and Guillain-Barré syndrome. So these are disorders that can occur and they're associated with a problem with the glial cells. So it kind of gives you an appreciation of the importance of the glial cells, even though they're not the ones that are sending nerve impulses or action potentials within the nervous system. And then we'll also talk about regeneration and what kind of regeneration, or to what extent we can have regeneration of axons in the peripheral nervous system versus in the central nervous system. Okay, so let's go back to our textbook. Okay, so the first nervous system disorder that affects the glial cell is called multiple sclerosis, or MS. And the glial cell that is affected is called the oligodendrocyte. And recall, that's the glial cell that's in the central nervous system, and its main function is to form myelin sheaths around the axon, so that way we get very, very fast action potentials traveling down the axons. So with this disorder, what happens is it's an autoimmune disease where the immune cells get a little bit confused and they look at oligodendrocytes as something that shouldn't be in the body, something foreign. And so they'll make antibodies against them and mark them for destruction. And those oligodendrocytes will be destroyed unless the myelin sheath is destroyed too. And that results in the ability to perceive your sensory information coming in, and motor coordination is impaired as well. So, and that's something that progresses as well. So that's a pretty, you can get permanent loss of functioning of some of the parts of your nervous system. All right, the other disorder is called Guillain-Barré syndrome, and this is a disease that also causes loss of myelin, but it does it in the peripheral nervous system, so around the peripheral nerves, and even around the roots of the spinal nerves that are coming off of the spinal cord. So it is muscle weakness that can progress to paralysis. And they don't exactly know the direct cause of this particular disorder. They have noticed that most people that develop this syndrome, they will have like a flu-like illness before they'll have the symptoms of the GBS. And so, but they never seem to be able to identify a particular bacteria or virus or anything that could have got that syndrome started and caused the inflammation with the myelin in the peripheral nervous system. So the person does usually recover almost all of the neurological function, and with just time, but it is a pretty scary disease because you don't really know why it happens, and it's obviously scary to have muscle weakness and even paralysis. So, but those are two of our disorders that you can kind of see what happens when the glial cells are not working properly. The neurons can't work properly, and then we'll get a lot of clinical signs related to it. Abnormality of the nervous functioning. So, all right. So I think the last thing that we're going to talk about is the regeneration of our peripheral nervous system axons versus the central nervous system. And I can't remember. Yeah, it's on the next page. All right. So the things that we want you to be familiar with. Obviously, the peripheral nervous system axons, any part of our nervous system is protected in the body, but it can be damaged. And so if we damage a peripheral nervous system nerve and the axons that are in that, we can have some regeneration, which means that we can get some grow back of that axon. But there are two factors that will determine if that's successful, if we get enough regeneration to actually regain function within that axon. So the first factor is the amount of damage. If there is like a severe amount of damage to that peripheral nervous system axon, then not as likely that we'll get complete regeneration and have complete return to function. So the amount of damage is going to be a big factor. And then the distance between where the axon was damaged to the structure that it is going to provide a nerve stimulation to, or an action potential to. So if that distance is really lengthy, If it's like a long distance, then there's going to be less possibility of regeneration and restoring the normal functioning. If it's a shorter distance, then it's more possible. So we will go kind of show you I don't ask you the steps on this, but it's kind of nice. I think I have a good picture. Yeah, kind of nice to see the regeneration and kind of what helps it regenerate. So here we have a random axon or random neuron here with its long axon going to, I guess that's supposed to be a muscle cell. Oh yeah, skeletal muscle fiber. So along the axon, if we have some sort of trauma, then again, depending on the severity and the distance from this trauma to the skeletal muscle, because that's what this neuron is innervating, will determine if it's going to regenerate. So we're going to say that trauma is not too bad and the distance is pretty short. So we're probably going to have some regeneration going on. So what helps with this regeneration is those little neurolymphocytes or the Schwann cells. They help guide the regeneration of that axon. So what they do is kind of form this kind of pathway, because they can undergo mitosis, remember, neurolymphocytes, Schwann cells, because they're glial cells. So they'll form this they'll undergo mitosis and form this nice little pathway that the axon can then regenerate along. And then again, over time, this is probably going to take longer than, days, probably take weeks or so, for that axon to regenerate and make its way to its skeletal muscle fiber in this case and establish a connection again. So this regeneration of the peripheral nervous system axons is possible. It does depend on the severity of the injury and also the distance between the injury and the structure that's being innervated by that neuron. But it can happen. Now, when it comes to the central nervous system, so brain and spinal cord, I think everybody's familiar with the fact that damaged neurons within the central nervous system is very limited. And there's a few reasons why. So in this case, this is where some oligodendrocytes are not helpful. Or some glial cells are not helpful, specifically oligodendrocytes. They can actually inhibit or stop axon growth because they produce these certain molecules. So they can actually kind of prevent that. And I'm not sure why they will do that, but that's one factor that prevents the regeneration of nervous tissue, specifically neurons in the brain and spinal cord. The other thing is when we're in the brain and spinal cord, that's where we have most of the neurons and they're all crowded in there, very tightly packed. So it's really hard for neurons to regrow just because of the spacing. There's just not a lot of space for them to regrow in there. And then the other thing that happens, again, it has to do with the glial cells. The astrocytes can form scar tissue. So they if we lose some of the parts of the axon or some of the neurons, what they do is lay down tissue, scar tissue, probably a lot of connective tissue. And then if they lay that down, or they stimulate that to be laid down, then there's no room for the axons to regenerate either. So the central nervous system is not likely to regenerate after injury. But they are doing a lot of medical research on this problem. So maybe sometime in the future we'll have some sort of therapy that would allow for regeneration of neurons within the spinal cord and brain, but for now, not likely. All right, so that is the end of our first 1 through 18 objectives for this week. So I'll see you in the next video for next week, and we'll start talking about the neurophysiology. So exactly how specifically our neurons are going to send action potentials and communicate and control the the body. So I'll see you in the next video.